Okay, so this is a list of all of the potential vaccines that are currently under development. They're being lumped together under this headline, the Warp Speed program at the moment. But each of these are things that the companies would like to develop. And these are the ones that are happening in America right now, but a lot of these are actually happening in multiple countries. And so most of these are multinational or international corporations. And so they'll be doing one arm of a trial in one place, one arm in another, and essentially using all those results together to try to figure out what works and what doesn't. But I think they'd rather figure out what works, really. So, each of these things is designed to deliver some part of the virus in a way that hopefully some part of the immune system would be able to see. And that's the challenge, trying to get part of the virus in front of the right section of the immune system. And to some extent, this differs in every individual person. So we all have genetically different versions of the MHC proteins, or in humans, those are called the HLA proteins. MHC stands for Major Histocompatibility Complex, and HLA is human leukocyte antigen, like human white blood cell antigen protein. All right, so people's responses will differ depending on their genetics and depending on the way that you put the virus in, or the part of the virus, and also depending in a large part on how efficiently you put the virus in, or exactly where. And there's a huge component of chance in all of this. Just because you put the right protein in front of the right person or the right person's cells does not necessarily mean that that person will generate a workable immune response to it. For any vaccine that's out there, there's a certain amount of failure where a person is definitely vaccinated and does not make a response. And to some extent, this is controlled by the host genetics. To some extent, it's controlled by the protein. And to some extent, we think it might be controlled by the way that you put the protein in. There are people that would tell you that, yes, definitely, the exact method of putting the protein in makes an enormous difference in the response. But what you find is that these experiments work really well in mice sometimes, not as well in people. And quite often, the differences that people see are not necessarily reproducible in other tests, in other labs, using what should be the same sort of equipment. So there's been a problem in figuring out how to standardize these tests. But what I want to do here is give you an overview of what we're talking about in terms of the vaccines that are out there. And we're actually going to start this week with what I think is probably going to be the first really good chance that we have to get a vaccine. I think the other ones may come through sooner, but I do not think they will give much of a response. Okay, so we'll start out over here from sort of high hype. We'll go down to low hype. And weirdly, the high hype and also contains the vaccines that I think have less of a chance of working. But that's my opinion. Now, it's based on a lot of published data for other versions, but most of these have not yet published any data whatsoever about how they work in the clinic. They're all in clinical trials of some sort. But there's no clinical trial data published. And so, yeah, big grain of salt there. We're working with the unknown, and it feels like, although this is logic, not science, but it feels like if these companies had something to crow about, they would come out and report their really massive results and just, dominate the market and get everybody's hopes up for a good reason. We're not seeing that, so either they're being very responsible and not doing that, or their results aren't great yet, but they're hoping they can tweak it and get better results. My guess is the second of those, but like I say, we'll find out when and if the data is actually published. Okay, so let's start over on the left side. So we've got the Moderna, the Sanofi, and the other Sanofi vaccine. And these are all kind of similar, and so we can cover them all together. So these are different ways to generate the spike protein of the SARS coronavirus 2. And Each of these uses a slightly different method to deliver the gene that encodes the spike protein, or the third one just, just delivers the spike protein right there, presumably by injection, although they haven't actually announced that as far as I can tell. So the ones at the top are messenger RNAs that encode the spike protein. So what's nice about this approach is that a messenger RNA is a temporary thing. So you're putting this gene in. It's not going to genetically modify the cells. It should make some protein from each messenger RNA that you put in, but it shouldn't be an unlimited amount of protein. And so, if anything, you're probably erring on the side of too little response or too weak an expression profile. But both of these companies, Sanofi Pasteur and Moderna, say that they have special compounds, special versions of lipids that are super extra good at delivering these messenger RNAs in, and they get massive, wonderful results that they nevertheless would love to show us but can't show us at this time. And hopefully they'll show us soon. Moderna is kind of famous for this. They do lots of experiments. They get venture capital somehow from people, but they never release their results. This has been kind of their MO as a company. So there we go. But Sanofi's a giant company, and so these are just different ways to put the spike protein all by itself in front of the immune system, and we're doing it quite directly. So in the Moderna and the first Sanofi method over here, the cells that get the messenger RNA will start to make spike proteins. Now, I think in both of these cases, the spike protein is missing the tail end of it. So it does not have the transmembrane region, or the region that was coded red on our first week's lecture that comes right before the transmembrane. And so these cells should just start releasing small amounts of spike protein into the bloodstream. And the idea is that there it would be picked up by antigen-presenting cells, things like neutrophils or macrophages or dendritic cells. And these cells would then chop it, which is called processing it, and pick out the right bits that match up with the proteins that they genetically have, and then go and show these to other white blood cells and get an immune response started. So, yeah, great idea. It's a thing that works outstandingly well in mice. And it ought to work in humans, but people have been trying this for years, and it has been problematic. It's one of those things where you can't really put a finger on why it doesn't work very well in humans, or it hasn't to this point, but yeah, it has not been great. Not been as good as it could be. All right, so that's the left side done. Let's go for the middle now. So in the middle, we have one from AstraZeneca, and this is a big European biopharma, and Johnson & Johnson, good old Johnson & Johnson. And the weird thing is they're essentially trying to do what looks like the same thing. So the slight difference is that, let me tell you what the thing is. So what they're taking is an adenovirus, and this is a thing that will get into your tonsils and cause them to swell up a little bit in some cases. But there are a lot of different adenoviruses, and they'll cause a mild fever or something like that. But generally, a few of them would cause something that you might call the common cold. But for the most part, they're just low-grade annoyance viruses for us. I don't believe there are many cases of lethal adenovirus infection. Although if we dig around, there's probably somebody who's immunocompromised in just the right way where this could potentially grow unchecked. So, first one is from AstraZeneca, and they're actually using the one that has, at least in the international press, been called the Oxford vaccine, or the Oxford adenovirus vaccine. This is very similar to a vaccine that came out of Oxford during the Ebola outbreak and was ultimately not successful in its first clinical trial and was put on a shelf. And the problem with it was that it did not generate enough of an immune response in people. And so there was another vaccine that came along that happens to look a lot like the one in the upper right corner. And it just did so well in its clinical trial that the Oxford vaccine got basically buried. But now that they have knocked out active Ebola cases, at least in that region, they're using the Oxford version of the vaccine to generate a little bit of an immune response. It makes an okay antibody response, not a great T cell response, kind of a low immune response overall. But what they're doing is taking the hollowed out shell of an adenovirus. Now, normally the adenovirus would contain double-stranded DNA that would start an adenovirus infection, and then you'd make lots more of these little shells. Instead of that DNA, they've replaced that and they put in the gene that codes for the SARS coronavirus spike. So now this thing looks like an adenovirus from the outside, and it gets into your cells the same way an adenovirus would, very easily. But it does not actually cause an adenovirus infection. It's a hollowed out version of this thing. And we will be talking about these, I think next week they start to come up. Let's see. The other one from Johnson & Johnson is essentially the same thing. Johnson & Johnson are using adenovirus 26. I forget which adenovirus the Oxford group are using. They call it chimeric adenovirus Oxford 1. Or it looks like Chad Ox nCoV 19, Chad Ox 1. Yeah, I have no idea how a human ever is supposed to pronounce that. But maybe it's a thing that's meant to be read and not said. So that's why we say the Oxford vaccine, because, yeah, I don't know. Chad Ox 1 nCoV 19 just doesn't have the same ring to it. And I believe it is a different adenovirus shell, but there are at least 40 different human adenoviruses, and you can essentially use any of the shells of these. But the risk is that if people have already had a particular adenovirus, then they will have made antibodies to it. And so if you're trying to deliver it in an adenovirus, on the one hand, this is good because the adenovirus can get right into your cells and deliver its payload just like it's supposed to do. But on the other hand, if you already make an antibody response against it, then you're going to neutralize the vaccine. So it's not going to work. Yeah. And so the companies that are doing these, the companies in the labs, these things all start out in the labs, that are doing these make a lot of modifications to the surface of the adenovirus. So even though it's mostly an old adenovirus that, comes from nature, they usually try to affect it in such a way that it would no longer look like an adenovirus to the average immune system. Now, there's going to be some immune response potentially if you've had this adenovirus before, but they're trying to minimize that, or to perhaps deliver enough virus quickly enough that it doesn't become a problem. But either way, they're both just delivering the soluble version of the spike protein, as far as I can tell. And they're using DNA to code the gene, and so it's going to go into the cell, and the cell is going to turn this from DNA into RNA. And the RNA will be a messenger RNA that will be translated to make the spike protein. So it's like one step longer than this, but it's basically doing the same thing. And that step happens automatically inside of any living cell. And so it's really no different or no more difficult. But as you can see, the first 5 vaccines that we've talked about are all... These are basically all putting their eggs in one basket. They are all looking at just the spike protein as the sole source of immunity. And this is also the case with the next 2 vaccines that we're going to look at, or candidate vaccines, I guess we should say. So over here on the right side, we've got the Merck live attenuated... that should not say, I copied that down. It's not an adenovirus 26. This is a vesicular stomatitis virus, so VSV, with the SARS coronavirus 2 spike protein in it. Now, Merck have announced this. They haven't published anything on it. There are papers that have been published on this, and one of those that is one that we're going to look at this week. But this is a technology that essentially any group could pick up. The sequence for vesicular stomatitis virus is public. Merck have previously made an Ebola vaccine that used regular vesicular stomatitis virus and chopped out the glycoprotein, the main glycoprotein gene there, and instead put in the Ebola equivalent of the spike protein. And so presumably they're just taking that thing, chopping the Ebola version back out, putting in the SARS coronavirus 2 spike, and then boom. And so this thing is a full-on live virus. It has all the other genes of the virus. It's a virus where you're essentially swapping the old spike protein that used to go on there for a new spike protein, which happens to be the SARS coronavirus 2 spike. So it should get into the same cells as SARS-CoV-2 spike, and you should be able to neutralize it with the same antibodies effectively. So they use these for neutralization assays quite commonly, but because they're safer to work with than the live SARS virus. You might be saying to yourself, well, how, if you put the spike from the coronavirus into there, why would we assume that this is not going to kill people the way that SARS coronavirus 2 does? And it's a good question, and it's a question that'll have to be sorted out in clinical trials. I was skeptical with the Ebola vaccine, and it worked fantastically well. The virus, Vesicular stomatitis virus, actually comes from a horse. And they're taking the version called Vesicular stomatitis Indiana virus, which is technically VSIV, but everybody just calls it VSV. And they are, genetically modifying that live virus and putting it in. The idea is this will deliver itself into the same cells that SARS coronavirus would. It'll grow with a lot of the same, setting off a lot of the same intracellular alarm bells that regular coronavirus infection would set off. But this is a particular virus that does not grow particularly well in human cells. So you would grow it up in something like horse cells or some kind of cell that's really good at growing this virus, which is fine. And then you put it into humans and the virus is just not great. It would have to make a ton of changes, and it looks like when it tries to make those changes, it just does not do well, and the virus basically kills itself through mutation if it tries to go that way. So, yeah. I think this one is promising. It's probably not going to produce too low an immune response. The risk is that it may produce too much immune response. And so this is going to be a Goldilocks kind of thing. You got your mama bear, you got your papa bear, and we got to figure out which one of these things, if any, is going to be the baby bear. Because we need this vaccine to be just right, for sure. Okay, the last one that I think is also very interesting, and both of these that are, the kind of live attenuated viruses, both of these are going to be coming through the corporation called Merck. And it looks like Mercs are going to be the ones that hit the immune system the hardest and probably have the greatest chance to produce the most immune response. The other one from Merck is from this other company, which I think might be French, but we'd have to look that up. What they're using is the Measles virus, which is going to be something that is actually similar in a lot of ways to vesicular stomatitis virus. It looks different, though, whereas vesicular stomatitis virus literally looks like a bullet bill. And on the outside of it, you're going to have those coronavirus spikes because that's where they go on the virus. This thing is more like just a bag. Yeah, a bag. And so on the outside of it, instead of these normal proteins, which I think are the F and G proteins, you're going to have the coronavirus spike proteins. F and H proteins. That's what they use. That's right. So, this is a strategy that has been tried with some success against Middle East respiratory syndrome coronavirus, which is similar in a lot of ways to SARS. We still don't have a good clinically proven vaccine, but this approach has been knocking around out there for a couple of years, and I think it may be in ongoing trials for MERS, but there aren't that many cases of MERS in a given year, and so they may be going for sort of slow and low trials over multiple years. That could be the case. So, once again, though, it is a live virus. It's going to go into a cell and replicate itself. And it just so happens that instead of just making the proteins that it normally makes, it's going to make also the spike protein of coronavirus. And it's going to produce this thing. And so that is another potential vaccine. And I might look at changing out one of the papers that we're talking about for this vaccine. We'll see. I don't know. We've got, I've tried to hit a lot of different approaches with the vaccine papers that we're going to cover, rather than doubling up on the same type of vaccine in any one of these. Because it's good to get a nice overview, I think, in a class like this. And most classes, honestly, that have anything to do with vaccine and antiviral development, These are things that go on, usually not at the master's level, but quite often at the PhD level. So I'm trying to, I'm trying to bring this to you in a way that would make sense given where you are right now and what you know. But yeah, a lot of this is, quite honestly, very advanced material. And you've done a great job so far in picking up on that and understanding it. So thanks. All right, so that's the end of this video.